Molecular markers. A molecular marker is a molecule contained within a sample taken from an organism or other matter. It can be used to reveal certain characteristics about the respective source. DNA, for example, is a molecular marker containing information about genetic disorders and the evolutionary history of life. Specific regions of DNA or genetic markers are used for diagnosing the autosomal recessive genetic disorder cystic fibrosis, taxonomic affinity or phylogenetics, and identity or DNA barcoding. Further, life forms are known to shed unique chemicals, including DNA, into the environment as an evidence of their presence in a particular location. Other biological markers like proteins are used in diagnostic tests for complex neurodegenerative disorders such as Alzheimer's disease. Non-biological molecular markers are also used, for example, in environmental studies. Uses of molecular markers. Molecular markers are used to study the taxonomic level of inquiry of population of species. They are used to study characterization of genetic diversity of species, its identification of adaptive trait loci using ISSRs, RFLPs, and allozymes, to study evolution of species by single gene or non-coding region whose sequencing was done using Sanger DNA sequencing, to study comparative genomics of species by sequencing multigene or whole genome by next generation DNA sequencing. Markers. Markers are of three types: biochemical markers, morphological markers, and molecular markers. Biochemical markers are of two types: isozymes and protein binding pattern. And molecular markers are either based on hybridization or on PCR or polymerase chain reaction. Examples of molecular markers based on hybridization are RFLP or restriction fragment length polymorphism, minisatellites and microsatellites. RAPD or random amplification of polymorphic DNA. Random amplification of polymorphic DNA or RAPD, pronounced rapid, is a type of polymerase chain reaction, but the segments of DNA that are amplified are random. The scientist performing RAPD creates several arbitrary short primers of 8 to 12 nucleotides, then proceeds with the PCR using large template of genomic DNA, hoping that fragments will amplify. By resolving the resulting patterns, a semi-unique profile can be gleaned from a RAPD reaction. They were developed independently by two different teams, Welch and McClelland, in 1990 and named RAPD and arbitrary primed PCR respectively. Protocol. RAPD involves isolation of DNA, deca-oligonucleotide enzyme, primer, tag DNA polymerase, keeping the tubes in PCR thermocycler, denaturing the DNA at 94 degrees Celsius for 1 minute, DNA strands separated, annealing of primer at 36 degrees Celsius for 2 minutes, primer anneal template DNA strands, DNA synthesis at 72 degrees Celsius for 1.5 minutes, PCR amplification and electrophoresis. Advantages. They are fast and economic. They have a relatively high power in detection of polymorphism. Primers are commercially available and do not require prior knowledge of the target DNA sequence, and it is easy to screen a large number of loci and individuals. Disadvantages. RAPD markers are dominant. Poor reproducibility. The marker is not locus specific. Homozygous and heterozygous are not easily distinguished. Applications. RAPD is used for species identification, analysis of population structure, analysis of genetic impact of environmental stressors, and analysis of genetic diversity. RFLP or restriction fragment length polymorphism. Restriction fragment length polymorphism or RFLP is a technique that exploits variations in homologous DNA sequences known as polymorphism in order to distinguish individuals, populations, or species or to pinpoint the locations of genes within a sequence. The term may refer to a polymorphism itself as detected through the differing locations of restriction enzyme sites or to a related laboratory technique by which such differences can be illustrated. In RFLP analysis, a DNA sample is digested into fragments by one or more restriction enzymes and the resulting restriction fragments are then separated by gel electrophoresis according to their size. It was invented in 1984 by the English scientist Alec Jeffreys during research into hereditary diseases. Protocol RFLP involves DNA extraction, digestion of DNA with restriction endonuclease, gel electrophoresis of the DNA fragments, transfer of DNA fragments using Southern blotting, hybridizing the DNA using labeled probe, and autoradiography. Advantages. The advantages of RFLP are that it is a codominant marker and it measures variation at the level of DNA sequence. Disadvantages. RFLP has a relatively low level of polymorphism. It is difficult and time consuming in development of markers to species which lack molecular information. It has a relatively low power in revealing genetic variation. It is labor intensive and requires relatively large amount of DNA. Applications. RFLPs can be applied in diversity and phylogenetic study. They have been widely used in gene mapping due to the ample availability of different restriction enzymes and random distribution throughout the genome. AFLP or Amplified Fragment Length Polymorphism. AFLP PCR or just AFLP is a PCR based tool used in genetic research, DNA fingerprinting, and in the practice of genetic engineering. Developed in the early 1990s by Keygene, AFLP uses restriction enzymes to digest genomic DNA, followed by ligation of adapters to the sticky ends of the restriction fragments. A subset of the restriction fragments is then selected to be amplified. This selection is achieved by using primers complementary to the adapter sequence, the restriction site sequence, and a few nucleotides inside the restriction site fragments as described in the amplified fragments are separated and visualized on agarose gel electrophoresis either through autoradiography or fluorescence methodologies or via automated capillary sequencing instruments. Protocol AFLP involves DNA extraction, digestion of DNA with restriction endonuclease, adapter ligation, preamplification, selective amplification, and gel electrophoresis. Advantages. AFLP allows the analysis of many loci simultaneously. High power analysis for revealing genomic polymorphism. High reproducibility does not require any prior molecular information and is fast, relatively inexpensive, and highly variable. Disadvantages. Markers are dominant and need special equipment. SSR or Simple Sequence Repeat. Simple sequence repeat is a microsatellite repetitive DNA marker, typically ranging from 5 to 50 times in a genome. The marker is highly variable. It is more similar to the short tandem repeats. They were discovered and developed by Litt and Lutti in 1989 and by Edwards et al. In 1991 in humans and were first applied to plants by Akaya et al. In 1992. Protocol. Here you can see the protocol of SSR. Advantages. SSR is codominant. Fewer high-quality markers are needed to strain differential. SSRs have stable amplification and good repeatability. SSR are easy to run and automate. Disadvantage: development cost and technical challenge for construction of enriched libraries for the species-specific primer. ISSR or Inter-Simple Sequence Repeat. ISSRs are DNA fragments of about 100 to 3000 base pairs located between adjacent, oppositely oriented microsatellite regions. Protocol: ISSR involves genomic DNA extraction, standardizing DNA quality and quantity. ISSR primer test, PCR amplification, protocol optimization. Now, if the amplificated bands are clear, then we can proceed with the next steps. Else, we'll need to exclude primer. The next step is to replicate with PCR amplification with optimized protocol. Then, if the bands are reproducible, we can proceed with the next step. Else, we'll need to exclude bands. The next step will be to record bands, genotype population samples, and then we'll score recorded bands and then analyze it. Advantages: the advantages of ISSR are: no sequence data for primer construction are needed. Low quantities of template DNA are required, randomly distributed throughout the genome. Disadvantages: the disadvantages of ISSR is that it is dominant marker and it has reproducibility problems. Applications: genetic identity, clone and strain identification, taxonomic studies of closely related species, and gene mapping studies. STR or short tandem repeats. A short tandem repeat in DNA occurs when a pattern of two or more nucleotides are repeated and the repeated sequences are directly adjacent to each other. The pattern can range in length from 2 to 6 base pairs or BP. For example, CATG in a genomic region. Protocol: the protocol of STR includes data collection, color separation, peak identification, peak sizing, comparison to allelic ladder, genotype assignment to alleles, peak editing to remove artifact calls, data review by analyst or examiner, and confirmation of results by second analyst or examiner. Application: used for the genetic fingerprinting of individuals. SCAR or Sequence Characterized Amplified Region. SCARs are DNA fragments amplified by the PCR using specific 15 to 30 base pair primers designed from nucleotide sequences established from cloned RAPD fragments linked to a trait of interest. By using longer PCR primers, SCARs do not face the problem of low reproducibility generally encountered with RAPDs. Protocol: the protocol of SCAR includes cutting and cloning the polymorphic band, sequence the insert and design specific primer, amplification of band of interest, re-amplification of the template, and seeing the clear pattern. Advantages: the advantages of SCAR are that it is quick and easy to use. It has a high reproducibility. It is locus specific and that it requires low quantities of template DNA. Disadvantage: it has a main disadvantage that it requires sequence information for designing the primer for SCAR. Application: gene mapping studies and marker assisted selection. EST or Express Sequence Tag. Express sequence tag is a 500 to 800 base pair subsequence of a short stretch nucleotides of the expressed gene or the cDNA. A small segment of the entire cDNA library is sequenced for the generation of EST. The EST marker is actually a short stretch of the individual cDNA, which represents as a tag for the entire cDNA. This tag can be used in the study and identification of the entire gene. There's no need to sequence an entire gene. Protocol: raw chromatogram, sequence and quality files, vector and low quality base trimming, check for chimeras, mask for repeats, merge repeats, clustering, assembly, contact or singlet processing, homology search, organism based annotation, and functional annotation. Advantages: EST is codominant. It is an efficient way to identify genes and analyze their expression by means of expression profiling. Useful for the development of cDNA microarrays. Great value in genome mapping. Disadvantage: identification of polymorphic ESTs is necessary for genome mapping. Application: identification of gene transcripts. Applications of molecular markers: DNA fingerprinting, construction of genetic linkage maps, population studies, identification of linked traits, and quantitative trait locus or QTL mapping. Thank you. If you like this video, don't forget to subscribe to get notifications for more new educational videos.